Good morning, guys. Can you hear me well?
Yeah.
Yes, sir.
Thank you. I made some problems, some practice problems for you, which are sort of representative of the problems that you might see on a test, on the first test, for example. And I put those on Canvas, right? So if you go to the quizzes section of your Canvas course site, then you will see that it says practice test problems. So please look at those, see how you can handle those. It will give you a good appreciation of whether you are studying right, whether you need to study more, whether you're studying correctly. And please, if you have any comments, put those comments in the discussion section of Canvas. Okay.
I had a quick question.
Yeah.
Since it is in the quiz area, is it going to be for a grade?
Well, it says practice there. So when I put it in the quiz area, I have a choice: graded versus practice. So this one will not go towards your grade now. And
okay.
I gave it, and you have multiple attempts, so you can try it as many times as you want.
Perfect. All right. Thank you very much.
And once you've done it, it'll tell you what the correct answers are. So, okay. So try that and let me know what you think. And as I mentioned to you, this is the first time I'm giving a test in using Canvas. So I'm learning as well. So any feedback will be appreciated. And let's go to the material. If you don't have any more questions, let's go share screen. Share screen. And we were talking about the alpha helices. Alpha helices, okay. So we talked about a variety of factors. So this is the alpha helix shown here. Remember, what holds it all together is these hydrogen bonds between the N and N+4 residue, right? So it will be the NH, amide NH of the N amino acid and N+4 amino acid. And so every NH, every peptide NH will be engaged in a hydrogen bond with the carbonyl. So that's what will hold the alpha helix together. Very strong cumulative effect. So, and there are some consequences in terms of what kind of amino acids are accommodated, what kinds are not. You can see here another big stabilizing factors is that these R groups, right? They all kind of perpendicular to the direction of the helix, and they all spread apart. Spread apart. So there is very little steric hindrance involved in the alpha helix. So another important factor which will favor the formation of the alpha helix. And you can see that actually the inside of the alpha helix, it's kind of squashed, right? So there's not much room. So very little can get inside. So these R groups on the outside, the amide hydrogen bonding is on inside, and it's pretty compact. So here are some numbers. Now these numbers you don't need to memorize, but some appreciation would be good, right? So 4 to 5 angstroms. This is the inner diameter. So too small for anything to fit inside. But the outer diameter is 10 to 12 angstroms. And with this kind of diameter, now this involves the side chains. So it fits well into the major groove of double-stranded DNA. So when we talk about DNA, we'll talk about major and minor grooves. And we will appreciate that proteins that regulate DNA function can fit nicely in between the base pairs. Not base pairs, but between the nucleotides. So Now there are proteins known as transcription factors. Anybody knows what these proteins are? Transcription. What do transcription factors do? So these are proteins which regulate gene expression, right? Depending on which gene needs to be expressed by a cell, a particular transcription factor will be activated, will go to the DNA, and will allow for the DNA to undergo transcription.
Is this like the ribosomes?
No, the ribosomes, remember, that's translation.
Oh, okay.
So the ribosomes, that's where you make protein,
Mm-hmm.
right? You start with transcription, you end with translation. Okay. So we also, if you go back to one slide here, so last time I kind of quizzed you on this plus and minus, and I think we got the answer. So this, what does this mean? So obviously plus next to the minus is good, right? What does it mean in terms of amino acids? What kind of amino acid do you want at position 1? And what kind of amino acid do you want in position 4? So let's say at position 1, I want lysine. What do I want at position N+4?
So why would you choose lysine?
Because it's positively charged.
Okay, so you want lysine to attach to position 1, or you want position 1 to be lysine?
Well, if lysine is in position 1,
okay,
then
it's positively charged.
Position 4, what amino acid will stabilize the alpha helix if I have lysine at this position?
Okay, so we would need something that's negatively charged?
Yeah.
Okay.
Give me something that is negatively charged.
Oh, okay.
Aspartate.
Aspartate. Good.
So could it just be like any of the positively charged ones and any of the negatively charged ones?
Right. So you have this situation where amino acid 1 is positively charged and it's negatively charged, or the other way around. So this will be a favorable primary sequence for the formation of the alpha helix. What about if I have something like this? Will this kind of sequence stabilize the alpha helix?
No.
And why?
They're both negatively charged.
They're both negatively charged. So this is something you have to look for. All right. So if I give you a sequence of, let's say, 12 amino acids, and I ask you, can you predict the secondary structure of that? So there are a number of things you can look for. So one is what is in these 1, 3 to 1, 4. It can be, because of 3.6 can be actually N+3 can be N+4, right? So because they're all going to be close, they're going to be on the same side. The charge is going to be close. So you look at what kind of charges are separated by 2 or 3 amino acids. And if they're opposite charges, that's going to be favorable. If they're the same charges, it's going to be unfavorable. All right.
So every first and every fourth is what we're looking at.
Right.
Amino acid. Okay.
Yeah. Well, so 4th and 5th actually. So these are, you see the 1 is kind of in between, right? So 3rd and 4th, right? So this is n+1, n+2, n+3, and n+4 will be the next one. So something like that.
Okay, I'm confused now. Okay, so to determine, okay, because you only put 4, you put, okay, like let's say the top one, the lysine, you have X, Y, and then you have the, what is that, aspartate?
You see, it's 3.6 residues, so it's not exactly it's not exactly the whole number.
Uh-huh.
So, and the reason for that is because you see the 4 is not exactly underneath of 1. It's 8, which is underneath of 1. So if you go n+7, right? So amino acid 1 and 8 will be directly in close contact. Amino acids which are 3 and 4, so this will be 3 amino acids away. This will be 4 amino acids away. So you look at those positions.
Okay, okay.
Okay. Now here's another question. What kind of sequence? You can see amino acids 1 and 8 lie nicely on top of each other. So if we go back, you see this is 1 and 8, right? So 4 and 5 will be kind of to the side slightly, but once you go through another term, then you align nicely. 8 will be aligned directly under 1. And that's because of the 3.6 amino acids. It's kind of, it's not 4, it's not 3. Yeah, but this is the nuance. We don't really need to worry about it. It's just getting too deep into this. What's important is that I want you to realize that what happens if you have 3 or 4 amino acid repeat. So what kind of sequence gives the alpha helix with one hydrophobic phase? So tell me. So let's say I have valine. What's my next amino acid? Something. What's my next amino acid? Something. What's my next amino acid? Now this one is important.
That one would have to come from the polar group, so like a threonine or something?
No, it has to come from the hydrophobic group. It has to be non-polar.
Oh, so they have to be, okay, so it doesn't have...
Because if I want one side of the helix to be all hydrophobic, right? I want to put all the hydrophobic amino acids on that side of the helix. So for example, I can put leucine there.
Could you put an aromatic there?
You can put aromatic. In fact, aromatics are good, especially when they're not just one aromatic, but several. For example, if I have phenylalanine and I have X, Y, and then tryptophan will be beautiful here. Okay. So aromatic amino acids interact with each other very nicely and form very nice hydrophobic phase. All right, so what else affects the helix stability? Okay, not all polypeptide sequences adopt alpha helical structures. So alanine, leucine are the best, right? These hydrophobic, small hydrophobic ones are great. Now, proline acts as a helix breaker because the rotation around the NC, shouldn't be A, should be alpha. NC alpha psi angle bond is impossible, right? Because now we have a ring and a ring will not allow for rotation. It freezes the dihedral angle in one particular place, right? It won't allow rotation. So, so proline, now I think you have the numbers here. No, maybe later. We'll look at the actual numbers. But so because the proline cannot rotate, it's a helix breaker. But there's another reason also. So I just told you the key to the stability of the alpha helix is these hydrogen bonds. So now if I draw the structure of proline in the amino acid, in the protein site, in the protein backbone, right? So there's one amide bond. There's another amide bond. What is another factor that will make proline destabilize the alpha helix? Think of hydrogen bonds. Remember, I want each of my amide NH hydrogen bond to be bonded to the N+4 carbonyl. Can this bond to the N+4 carbonyl? Yes? Do you see hydrogen on this?
Are
No,
you
the
asking if
hydrogen.
it can make a linkage?
Yes, the hydrogen has been taken away. There's no hydrogen. Instead, we have an alcohol.
Yes. Yes, so it can bond.
There is no hydrogen bonding. So this,
Oh, you're saying, I thought you were saying, can it connect to something?
no, because there is no hydrogen, then there will be no hydrogen bond. And remember, it's the hydrogen bonding that keeps the alpha helix together. And if you have too many prolines in a sequence, prolines will not be able
Oh,
to.
you're saying from what the question?
Yeah, prolines will not be able to stabilize the alpha helix through hydrogen bonds because they don't have those. Now, another one that is a helix breaker is glycine. And why do you think the glycine? Well, here's the answer. The tiny R group, which is a hydrogen, supports other conformations. Remember, I told you one of the reasons why alpha helix is so good, why it's so favorable, is because all these R groups, there's no steric hindrance, right? They all spread apart in different directions, no steric hindrance. And now if you have a whole bunch of these glycines here, instead of the amino acids with large R groups, then there's no gain in energy, right? There's nothing to spread apart. And so the glycine-rich segment may adopt some other conformation because there's no benefit in having the alpha helix. And we just talked about attractive and repulsive interactions between the 3 to 4 amino acids apart that will affect the formation. And there's, you can see here, now I don't want to memorize these numbers, but these are the energy gains. So if the alanine is considered to be the best to form the alpha helix, you can see here, so this is the less energy gains, smaller energy gains. You can see here arginine is very close, but that again depends on what's in the N+4 position, right? So this is just the average number, 0.3 delta kilojoules per mole. But what if you have aspartate in the N+4? Then this number may not be 0.3, it may be even negative, maybe even better than alanine. And you can see glycine is really bad, right? Glycine is 4.6. So there's very little energy gain through forming alpha helix, and proline is bad too. And the other amino acids are somewhere in between. Make sense? And the last factor which affects the stability of the alpha helix, it's what's known as a large macroscopic dipole moment, okay? So this results from the fact, so for amino terminus, you have a NH3 plus, carboxyl terminus, you have carboxylate, COO minus, right? And so there's a dipole moment with a negative charge in the carboxyl region and a positive charge in the amino region of an alpha helix. And so the primary sequence will help stabilize the alpha helix if you have negatively charged amino acids accumulated at the amino terminus and positively charged amino acids near the carboxyl. Right? So for example, if I give you a sequence and you will see like there's a whole bunch of lysines, arginines at the carboxyl end, right? And a whole bunch of aspartates and glutamates near the amino terminus, you may tell me, oh yeah, I think this will adopt the alpha helix because the amino acid sequence stabilizes the macroscopic dipole moment of the alpha helix. Make sense? Any questions?
I have a question.
Yeah.
I have a question. So are you talking about the amino terminus and the carboxylic terminus of any amino acid, or is this a specific amino acid?
So you're asking what the carboxyl terminus and amino terminus are?
Yeah, basically. Just on that.
Sorry, repeat it again.
I said basically you can explain it. Yeah, I mean.
Yeah, so remember that when we do the sequence, when we draw a structure of a peptide, the amino terminus will always be on the left, carboxyl terminus will be on the right. And what you have to do, you have to look at
I'm saying like, okay, go ahead.
And you have to look at these amino acids here, and you have to look at these amino acids here. Right? Okay. Beta sheets. All right, so this is the second secondary structure we're going to look at. And so here, because of the planarity of the peptide bond, it's possible to have some type of pleated sheet-like structures, okay? And they're held together by hydrogen bonds between the backbone amides and different strands, right? So in the alpha helix, it was the same strand, right? It's like this. And the hydrogen bonds were like that, like that, like that. And here, it's going to be different. So let me give you an example. So parallel, antiparallel beta sheets. So sheets together by hydrogen bonding of amide carbonyl groups, opposite strands. Okay, so here, what we need to look at is parallel and antiparallel. So this will be parallel. And parallel will result from some kind of disorientation. Let's say you go this way, then some kind of random coil comes back and goes in the same direction as the original strand. And then there will be hydrogen bonds in between these strands. So this will be parallel. And you can see that now it doesn't have to be, but this is the same strand. It's just it just goes away, does something else, adopts some other kind of secondary structure. Could be alpha helix, could be couple alpha helices somewhere. Comes back, aligns in a parallel direction, and forms a bunch of hydrogen bonds with the original strand, I mean, with the original portion of the strand. So the way it works, you can see here. So this is parallel beta sheet. Now here's the amide bond, right? Amide bond, amide bond, amide bond, and you can see what happens. They alternate. So the two amide bonds come close to each other, and so the carbonyl of the second one will serve as the hydrogen bond acceptor, and the NH will serve as a hydrogen bond donor. And then they switch roles. And in the next amide, this carbonyl is a hydrogen bond acceptor, this hydrogen bond donor. Donor, acceptor, donor, acceptor, donor, acceptor. And this the opposite on this one. Acceptor, donor, acceptor, donor, acceptor, donor. Now, these are somewhat weaker hydrogen bonds because they are at an angle. Remember, hydrogen bonds like to be on one straight line, but because there are so many of them, it's a pretty strong secondary structure, right? And you can have a variety of these strands coming together from, for example, this one can go somewhere for a walk and then come back. And then come back. Like so. And then you have hydrogen bonds here. All parallel, right? So this will be a beta sheet.
Where are beta sheets usually seen?
So we'll talk about, so there are some structural, because they're pretty strong as well. So there's some structural proteins. Actually, we'll look at some examples later on, like silk protein, for example. And we will look at some globular proteins which are in solution. There we'll see that actually alpha helices and beta sheets, they are predominant secondary structures in water-soluble globular proteins, such as antibodies or myoglobin, which is the oxygen transport protein. So basically anything that is water-soluble. But we will see just maybe even in this lecture, yeah, we should see it in this lecture. And now the second one is anti-parallel. So now you can see that actually this goes in this direction and this strand goes in that direction. So usually the way this works is you have a sharp turn and this goes back. And then, so this goes this way, this goes that way. And usually this will be then, when it's anti-parallel, it's the same strand, just with a sharp turn and going back, okay? So here, there's a different type of hydrogen bonds. Again, it alternates. Hydrogen, now NH, carbonyl, carbonyl, NH. NH, carbonyl, carbonyl, NH. So, and here the hydrogen bonds are linear, right? So it's somewhat so stronger. But again, they're nicely positioned to be, to have a repetitive character, right? And here we have donor, acceptor, donor, acceptor, donor, acceptor, and the opposite on this one. So this will be anti-parallel beta sheet. So these are somewhat stronger. In fact, when we talk about, let's see. When we talk about protein fibrin, which is the basic structure of a spider web. So there we have anti-parallel beta sheets. And anti-parallel are again stronger than parallel because of the nature of the hydrogen bonds. And that's why spider web, for example, is so strong, right? Because anti-parallel beta sheets are stronger than parallel beta sheets. All right, and then you may want to ask, what kind of structure is here? So these are known as beta turns. So that's the third type of secondary structure that we're going to look at, beta turns. So frequently, whenever strands in beta sheet change the direction, that's what I just drew for you. So the 180 turn is accomplished over 4 amino acids. And so the turn is stabilized by a hydrogen bond from a carbonyl oxygen to amide proton 3 residues down the sequence. And here is where proline and glycine are actually favorable amino acids, right? So in alpha helix, we didn't want proline and glycine. In beta turns, that's exactly what we want. So here is, there are 2 types of beta turn. Don't worry about the difference. The difference is just kind of puckering. The difference is actually type of twist. But the whole point is that you come in from amino acid 1, and there's a carbonyl here. Then there is a proline residue at this position. Here, usually glycine is favored. And then the NH of the amino acid 4, that forms a hydrogen bond here. That's what locks the beta turn in place, right? So the carbonyl of amino acid 1 and the NH of the amino acid 4. And that's the same in type 1 beta turn and type 2 beta turn, carbonyl and NH. So the only difference is actually how it's arranged, how this bridge is arranged, right? So here we have the carbonyl kind of going away and nitrogen towards us. Here we have the carbonyl coming towards us, nitrogen going away. So it's just a different way this bridge kind of folds into place. So don't worry so much about the difference. I will not focus on that as much. Just be aware there are 2 types, type 1 and type 2 beta turn. And so what's important here is obviously is the ability of the proline. You can see here, you want this to be able to make a sharp turn. And so as we talked about, most amino acids, they like to adopt the zigzag 180-degree conformation, right? And proline is the only amino acid which actually doesn't really care so much. So you can see here, most peptide bonds not involving proline are in trans configuration, 99.9. But for peptide bonds involving proline, about 6% are in a cis configuration. And most of the 6 involve beta turns, right? So most beta turns will have a proline that is in a cis configuration, and there's an enzyme, proline isomerases. So let's look at that. So here's our trans, right? So this is proline. You can see here, this is the omega, the hydro angle, and there's a partial double bond character. And the angle here is 180 degrees. So it's trans, right? This goes in this direction, and this goes in this direction. So it's trans. Now then there's the enzyme comes in, proline, isomerase, which will make this rotate about this partial double bonded bond, which has partial double bond character. And here now we have a cis configuration. So this is in, and this are in the same direction, right? So again, partial double bond character. And the angle here is 0 degrees, and it's cis. And so 6% of all prolines in proteins are in cis, and they're particularly found in beta turns. So this is what allows for a strand to make a sharp turn, sharp 180-degree turn, and go in the opposite direction. This makes sense? Any questions?
Makes sense.
Yeah. Makes sense to me too. It's pretty straightforward. Just keep in mind the proline, trans, and cis. Most amino acids are in trans, but proline can easily adopt cis. There's an enzyme that does that. And these kind of conformations occur in the beta turns. So these are 3 secondary structures we want you to know, right? Some details about them. And so the question is, when you study proteins, obviously, we'll talk a bit about X-ray crystallography. We'll talk about the NMR, nuclear magnetic resonance spectrometry, how those are used to deduct the secondary structures. In the past, the most classical way actually was CD, basically circular dichroism. Circular dichroism. It's CD analysis. Basically, what that is that, remember circularly polarized light, right? So we talked about that with optically active compounds in organic chemistry too. No, sorry, there we talked about plane polarized light. So this is circularly polarized light. So basically just different type of light, but it's polarized. Again, it's not in all possible directions, but instead it's kind of a spiral. It's more physics. So now you can do it in left and right, just like what I showed to you. If it's clockwise, it's right. If it's counterclockwise, it's left. And so this is the extinction coefficient. This is for the left circularly polarized light. This is for the right circularly polarized light. And if you take the difference, you get this delta, which is the difference between the left and the right extinction coefficients. And you can get the signal based on that, okay? And there is characteristic signals. So let's look at that. And so this is delta extinction coefficient, delta epsilon, and this is the wavelength, right? And so depending on the wavelength, this difference between the L and R will change. And if the curve of the graph is like this, starts at the top, drops to the bottom, flat a bit, and then becomes 0 at longer wavelength, then you're pretty certain that you're dealing with alpha helix. If it's like this, the difference is smaller, and then again comes back together at 0, then it's beta conformation. And the random coil, when there's no particular secondary structure, this actually is negative, the difference is negative, and then flattens out. So if you get one of these 3 graphs, then you're pretty certain what kind of structure you're dealing with. The only problem is, as I mentioned to you, many proteins have both, or have actually different kinds of secondary structure. And so in most cases, this is kind of messy, unless you're dealing with a protein or protein segment, which has only one type of secondary structure, right? Then you get a very clear picture, very clear curve that you can use to assign the secondary structure. Okay, so the circular dichroism. Now the reason for that kind of curve, I'm sure it's been investigated. I don't know if somebody has answered that question. It's obviously started as just an experimental observation, but I'm sure it's studied by theoreticians, and I don't know what the final word is, if they know the reason for that or not. Maybe. All right, so these are 3 kinds of secondary structures. Now we move on to tertiary structure. So remember, so now the tertiary structure, we're going to deal with the entire protein, not just a portion of the protein with a particular secondary structure, but the entire overall spatial arrangement of atoms in a protein. Now, so these will have various secondary structures which are stabilized by numerous weak interactions. So they'll be interacting with each other. And the obviously amino acid side chains will be interacting with each other. Hydrophobic, polar, and many of them will be stabilized by disulfide bonds. So we talked about those. Right, so cysteine, amino acid cysteine, if you oxidize the sulfhydryl group, you form a disulfide linkage. Okay, and interacting amino acids not necessarily next to each other in primary sequence, right? So you can have a protein segment, then random coil, maybe even 100 amino acids. It goes for a walk somewhere, comes back as a beta sheet, and this beta sheet will interact with the alpha helix that was 100 amino acids before, right? So two major classes of proteins. There's fibrous and there's globular, okay? So for fibrous, so let's see, what can we say in general about fibrous proteins? So for fibrous, let me maybe just write it here. Fibrous. So usually one type of secondary structure. One type of secondary structure. And one main reason is that one main function provides support. Provide support.
So you're saying fibrous is not a tertiary structure?
It is a tertiary structure, but in most cases, it's just, even though it's a tertiary, well, when we talk about tertiary structure, we're talking about tertiary structure of the entire protein, right? So the entire protein can have just one type of secondary structure or many different kinds of secondary structures in them. So fibrous proteins usually contain one type of secondary structure.
Okay, got it.
So for globular, many types of secondary structure. And in most cases, these are going to be water-soluble proteins, and most of these will be enzymes. Or what's known as a regulatory.
Which one's water and which one's lipid soluble?
You know, that's probably an incorrect. Well, so globular mostly water soluble, but no, it's not like one is water, one is lipid soluble. So it's ignore this. It's it's not that fibrous is water and globular is lipid soluble, right? So there's no connection between these two statements. Okay. So some of these are water soluble, some of these lipid soluble, and it can be either one. In general, fibrous proteins are not really soluble in anything because they're like rods. They're they allow for very little conformational change to express or to decorate themselves with polar residues on the outside, right? So the fibrous proteins are not capable of presenting their polar groups on the outside surface for water solubility. Now, globular are just the opposite, right? So many enzymes are water soluble. Although there are enzymes which are lipid soluble too. All right, so here is fibrous proteins from structure to function. So we will look at alpha helix, right? So here we'll look at alpha keratin. So the protein which is based on alpha helix. We'll talk about beta conformation, silk fibroin, I don't know how to pronounce it properly, and collagen triple helix. So this is the protein for tendons, cartilages, bone matrix. All right. So we'll look at three types of, now these are, even though these are tertiary structures, but because they are present throughout the entire protein, so we can also refer to them as secondary structures. So here, secondary and tertiary structures are basically the same thing. All right, so this is a hair. Now, so keratin is a component of a bunch of strong.
Can you go back to the last slide real quick?
Yeah. Yes.
Okay, you can go ahead.
Okay, so keratin, the component of, so obviously hair. In fact, we have a hair right here in front of us. Component of hair, nails, claws, quills, horns, hooves, basically anything, even the outer layer of skin. Outer layer of skin. Now, so these are cells, right, within the hair. And you can see actually they're composed of what's known as intermediate filaments, right? So this whole thing, which sticks out from one cell, this will be an intermediate filament, which composes simpler structures, protofibrils, protofilaments, and eventually it comes down to two-chain coiled coil. So keratin, what it represents is actually, it's a right-handed helix. Right-handed helix. Let's see what we have on next. Yeah, I think maybe it's easier to look at this slide. So it's a right-handed helix. You can see if you stand in this direction, and the direction of propagation is such it's going to be clockwise. Right-handed helix. And the two strands of the right-handed helix, they are wound around each other to form left-handed helix. That's why it's called a two-chain coiled coil, right? So two coils, and each coil is coiled. So
does the left-handed coil, is that the protofilament?
So this will be, so each coil is the right-handed helix.
Right.
Right-handed helix. Which is and both of these helices form another coil, which are wound around each other in left-handed mode. What's the right way to say that? Just one second. So right-handed helix or helices, interwound in left-handed fashion.
Is that left-handed super twist? Is that what you mean?
Well, I am not exactly sure what super twist is. What's super twist?
I don't know. It's in the book, and I think it's the same thing. I didn't read it all, but I think that's what it means. Okay.
Yeah, I mean my knowledge of these twists is pretty limited. So super twist will be like a tight twist, or anybody knows what super twist means? Maybe we can talk about this in a discussion. Left-handed, wound in left-handed mode. Left-handed mode. I mean, that's how I understand it, right? So right-handed helix, but relative to each other, they are left-handed, right? So
And what does that mean?
maybe super twist. Maybe super twist exactly what that means. Coil. Maybe coiled coil means super twist.
Why is it named by right-handed and left-handed?
That's what I was telling you about, the clockwise versus counterclockwise turn. Right? So you can see if you look in this direction, or it doesn't matter which direction you look, it's going to be the same. Direction of propagation along the helix is going to be clockwise. So it's going to be right-handed helices interwound in left-handed mode. Right, and then, so basically these then aggregate together to form protofilaments, protofibrils, and eventually intermediate filaments. So intermediate filaments are components which are responsible for the strength of the hair, but they also responsible, for example, for cellular cytoskeleton, right? So remember I told you that cytoskeleton in the cells, there are 3 different types of structural components. There is actin cytoskeleton, there is microtubules, and there is intermediate filaments, right? So intermediate filaments are responsible for the shape of the cell, are responsible for the change in the shape of the cell, and the strength. So the cell can adopt particular shape and not easily change that shape, withstand various osmotic pressures. All right, so this is keratin. Now there's keratin is also the strengths, which are the helices. They're stabilized, they're drawn to each other through disulfide links. Okay. In fact, so obviously the stronger the disulfide bonds, or the more you have disulfide bonds, the stronger the keratin. And so I told you that the rhinoceros horn is made of keratin. And so in rhinoceros, %, of cysteine amino acids are engaged in disulfide bonds. There is a small box in your textbook that talks about the chemistry of permanent waving, right? So if this is your hair, and let's say, how do you change the your hairstyle, right? So there's all these hydrogen bond, so all these disulfide bonding linking the keratin strands together, right? And then what needs to be done before you apply the heat and moisture, you need to break the disulfide linkages. So you treat that with a reducing agent, right? Reducing agent, which usually contains the sulfhydryl group, which will break up disulfide linkages into cysteines. Then you curl it, right? So it will change the shape, and then you reoxidize it. So these thiols or the cysteines will shift relative to these cysteines, and now you change the order in how these cysteines form disulfide linkages, right? So they were ordered in this way. Now there is basically, you switch the positions, and so some of them can form disulfide bonds, some of them cannot. And so you can change the orientation and change the hairstyle in more or less permanent way, right? Until you apply this again, because of the strength of the disulfide linkages.
So this is what I'm gonna be thinking about every time I straighten out my hair. That's pretty cool.
Yeah, when you apply the reducing agent, you know exactly what you're doing there. You're breaking the disulfide linkages. And then when you apply the oxidizing agent, what do you apply at the end? I don't know, because I've never done it.
To bring it back?
Yeah.
Water.
Don't you apply some kind of oxidizing agent?
No. If I wash my hair, it just, I don't know what's in the shampoo and stuff. If I wash my hair, it gets wavy again.
Okay. All right, let's move on to collagen. So this is another protein which has evolved for strength. This one is found in connective tissue. So tendons, cartilage, and bones, cornea of the eye, okay? And so again, here it's the two prominent amino acids are glycine and proline. You can see how some groups of proteins love glycine and proline, and some groups hate it, right? And this will be actually left-handed helix. And 3 collagen chains intertwine into right-handed superhelical triple helix. And so we'll look at the structure in a moment. Superhelical triple helix. The triple helix has a higher tensile strength than a steel wire of equal cross section. This is how strong collagen fibers are. And they assemble into collagen fibrils. So we'll talk about that in a moment. So let's go back. Right-handed superhelical triple helix. So the left-handed helix, right? So here we have a, so this is the structure of collagen right here. So we have a left-handed helix. So if you can see here, direction of propagation, right? This is going to be counterclockwise, like that, like that, like that. Counterclockwise, so it's a left-handed helix. And so three of these are put together and form a right-handed triple helix. And the main amino acids which form the left-handed helix in collagen are glycine, proline, and 4-hydroxyproline. So we talked about 4-hydroxyproline as an amino acid that is not one of the 20 amino acids, but it's an amino acid which undergoes oxidation, and it becomes an important component of a protein such as collagen.
Sorry, could you repeat what the three of them form again?
So they alternate. So 3 amino acids residues per turn. Okay. And these 3 amino acids in most cases are glycine, proline, and 4-hydroxyproline. Is that what you asked?
I meant the three left-handed helix. It forms a right-handed helix.
Right-handed triple helix.
Triple. Okay. Thank you.
Right-handed. Right-handed triple form right-handed triple helix. Okay. So each turn, so there are 3 amino acid residues per turn, right? And these are the 3 amino acids which are present. 4-hydroxy, proline. Hydroxyproline. Now, all 4 are important. Okay. Oh, sorry, all 3 are important. Now, glycine is the key. If you don't have glycine, actually, we'll look at there are some diseases because of the mutation, because of genetic diseases, because glycine has been replaced by some other amino acids. Glycine is very important because if you think about this triple helix, right? So there is an internal side. So in other words, the triple helices, they come together, and there's very little room inside of that triple helix, right? It's very hard to accommodate the side chains. And so glycine is a key amino acid because it doesn't have any side chain. And so without glycine, it's going to be very hard to form a triple helix like that. So glycine is key. Glycine is key. Because of tight junctions. because of tight junctions between individual chains. Individual chains. Now, this 4-hydroxy proline in collagen. Now, very important. So the reason for why it's important, it forces the proline ring into a favorable pucker. So if you go to the next slide here, you can see here. So proline without the hydroxy group is shown right here, and it's known as endo. It's known as endo because this carbon here and this carbonyl are close to each other. Remember, endo is on the same side. If you put a hydroxy group here, it changes the pucker, basically changes the orientation of the 5-membered nitrogen-containing ring. And so now this carbon and this carbonyl are away from each other, and that's known as an exo conformation. So the hydroxy group is crucially important. And there's an enzyme. There's an enzyme known as prolyl hydroxylase. Prolyl hydroxylase, which introduces this hydroxyl group onto the collagen, right? And it's an example of what's known as a post-translational processing or post-translational modification. Remember, I asked you last time what that meant. So after the protein has been translated on the ribosomes, then the enzyme comes in and puts on that hydroxyl group on the protein. So the enzyme requires alpha-ketoglutarate, molecular oxygen, and ascorbate, vitamin C. It requires vitamin C. The left actually is empty page here for us to write down a few things. Who knows what scurvy is? Scurvy.
It's a lack of vitamin C, essentially. It's a disease caused by a lack of vitamin C.
A lack of vitamin C, and what are the symptoms?
I know bleeding gums is one of them, but I don't know the rest.
Yep.
Yeah, like just weakness, I guess.
It causes like a breakdown of the collagen, right? So it'd be anything, just, I mean, throughout the whole body, you'd have symptoms.
Yeah, basically anything that utilizes collagen will collapse. Bleeding gums, blood vessels which contain collagen, so causes hemorrhages. I hope I spelled this correctly. Hemorrhages due to the weak blood vessels or fragile blood vessels. Tooth loss, basically, and eventually all these, if you continue to live without vitamin C, all that will lead to death, right? And you know, this was the disease was originally characterized as disease of people who were engaged in long sea travel, right? So like crusaders, for example, traveling to Egypt, for example, right, to spread Christianity. So many of them would get the symptoms of scurvy. And even nowadays, if you think about, there's been a lot of studies on even not just in remote areas where people's diet did not include vitamin C, but even on student colleges, on college campuses, right? There have been many studies showing that students don't consume enough vitamin C, and many of them have mild vitamins of scurvy, and some of them have a pretty severe signs or severe symptoms of scurvy, right? So you should eat your fruit and vegetables. Eat fruits and vegetables. That's the main source of vitamin C. You know, most mammals biosynthesize vitamin C, but humans and some other mammals, such as various types of monkeys and some other animals, have lost the ability to biosynthesize vitamin C. And we have to obtain it through the diet. One apple away does what? No, one apple a day
Keeps the doctor away.
Keeps the doctor away, right? Eat your vitamin C. All right, so I told you what this does, this hydroxy group, right? And specifically, what does ascorbic acid? So vitamin C is the same as ascorbic acid. Vitamin C. So, so if this is the proline residue, right? So we're gonna oxidize hydroxylate at this position, at this carbon. And so there's a starting material, alpha ketoglutarate and molecular oxygen, and we utilize the iron on the enzyme. The enzyme is prolyl 4-hydroxylase. So the enzyme will hydroxylate this position, produce carbon dioxide as a side product, and succinate will result from alpha ketoglutarate. But there's also a second cycle here of the enzyme, which utilizes alpha ketoglutarate, molecular oxygen, and ascorbate. So what happens here is that if it wasn't for the ascorbate, then the iron 2+, remember iron goes back from iron 2+ to iron 3+, back and forth. So the second cycle of this enzyme will, if the iron 2+ undergoes oxidation to iron 3+, we need to bring it back, right, to iron 2+, and that's what the ascorbic acid will do. Now, I think there's a mistake on the slide. I think this should be iron 3+. If this is iron 2+, the product is iron 3+. So iron undergoes oxidation. So then iron 3+ enters the second cycle and regenerates iron 2+. And that's what ascorbic acid is necessary for, is to regenerate iron 2+ that can then go back and participate in the next cycle, in the next cycle of the enzymatic action. Like that, right? So, and vitamin C is the reducing agent, which will undergo oxidation to dihydroascorbate, shown here. right? So it restores the iron 2 plus state. So obviously without the vitamin C, all the iron will be oxidized to iron 3 plus, and it will no longer be available for the first cycle where the introduction of the hydroxy group takes place. Any questions? All right, and so collagen also these triple helices of collagen, they also interact with each other, forming collagen fibrils, and many times they're cross-linked, and they form extremely strong superstructures. Extremely strong superstructures, stronger than like metal and things like that. Now there are some mutations, right? So collagen, so there are some diseases, genetic diseases. It's important to realize. So there is a disease called osteogenesis imperfecta. And there's another one called Ehlers Danlos syndrome. Ehlers-Danlos syndrome. So both result from the genetic mutation result from glycine substitution by other amino acids, for example, cysteine or serine. Cysteine or serine. So the side effects are, the symptoms are, for example, things like loose joints and fragile bones, loose joints, right? Some horrible diseases. For example, there's a composer, Niccolo Paganini. If you've heard of him. Paganini, who had one of these diseases. So, and the last one I want to mention is silk fibrin. Now, so this is the main protein in silk from moths and spiders. So based on antiparallel beta sheets, remember I told you antiparallels are stronger than parallel. Small side chain, alanine, glycine allow for the close packing of sheets, and hydrogen bonding with each sheets, London dispersion interactions between sheets. And here's so basically these are, sheets which are packed one on top of another, right? And you see the antiparallel. If this is towards us, away from us, towards us, away from us, and so on and so forth. And is there anything else I want to say about it? Yeah. So one thing to keep in mind is that, remember, I told you that alpha helix can stretch quite a bit. Beta sheets cannot stretch. They're already in a fully extended conformation. Yeah. So if you take spider web, you will see that you try to stretch it. At some point, it will just simply break, but you will not be able to stretch it. In fully extended conformation. All right, let's see. I think that's all I wanted to say. Are there any questions about the material today? On Monday, it's going to be our last lecture on this chapter. We'll talk about globular proteins. We'll talk about intrinsically disordered proteins, proteins that don't have a permanent three-dimensional structure. We'll talk about X-ray crystallography a bit, NMR. And we'll talk about how the proteins actually find their native folds, right? So far we've looked at the folds that already exist, right? But the question is, how do they find these native folds, right? Once they come off the ribosome, what are some special proteins that help them find these native folds? And what happens to misfolded proteins, right? And what kind of diseases come from protein misfolding? All right. So if there are no questions, I will talk to you on Monday. Try the practice test and let me know what you think. Bye.
Thank you. Have a good one.
Thank you.
You too.